Morning students, I am Mr. Tajendra again, and today we will talk about the adaptive immunity. In the previous lecture, we already discussed about the what is the immunity, what are the types of immunity, what is the innate immunity. Okay, so we discussed that immunity is of mainly two type: innate immunity and acquired immunity. And today we will discuss about the adaptive immunity or acquired immunity. Okay, so because already we discussed about the innate immunity, so today's our topic is about adaptive or acquired immunity. So this is the second part actually. So adaptive immunity, or we can say that it is acquired immunity. Okay, so adaptive or acquired immunity is based on the two activities, and that are cell mediated immunity and humoral immunity. Okay, so how it is possible, and what are the main types and how it works, we will discuss about all these things. Okay, so first of all, as I told you in the previous lecture, that immunity is of depends upon the many line of defense, and I told you that first and second line defense are mainly related to the innate immunity. Okay, but third line is based mainly related to the adaptive immunity. Okay, so not only third, second line also plays some role in the third in the adaptive immunity as well, and this adaptive immunity is also related with the second line. Okay, it means third and second line both plays a role in the adaptive immunity, but main or major role is of third line. Okay. Joint and overlapped with the innate immunity. Adaptive immunity itself is not separated from anyone else. It is also related to the. It is also adjusted with the, or we can say that it is also dependent on the innate immunity, right? So adaptive immunity is not is independent. It is waiting for the some signals. It is always waiting for the some signals which are provided by the innate immunity, and then after these receiving these signals, it starts working. Okay, so whenever we are talking about the adaptive immunity, then it contains, as I told you, that cell-mediated immunity and humoral immunity. So cell-mediated immunity, it contains B lymphocyte, T lymphocyte, and their memory cells. Okay, so that is the cell-mediated immunity. It is a part. It is a part of an adaptive immunity, and the second part is the molecular, and that contains the growth factor, cytokines, cell surface ligands, proteins, chemokine, and soluble immune factors or proteins like antibodies. Okay, so these all are mainly part of the adaptive immunity. Okay, these are parts of the adaptive immunity. Humoral immunity, which is mainly based on the or rely on the B lymphocyte, whereas cell-mediated immunity, it mainly relies on the T lymphocyte. Okay, because humoral itself mean with the soluble, okay, soluble type of proteins they contain. Okay, whereas cell-mediated itself, its name suggests that there are main involvement of the cells are there. Okay, so that's why we can say that there are T cells, because T cells always produces some more type of cells which can help in uh, creating the immunity in the form of cells, whereas the uh, immunity is provided in the form of the proteins and soluble type of, or in soluble and sometimes in non-soluble, insoluble also. Okay, so there are some proteins or some type of factors, molecules which are biochemicals and released which are releasing by the uh, cells of the immune system, and these cells are very important in the humoral immunity. Okay, so important example is like Ig, or we can say that antibody. So important is the antibody. Okay, and some are complement system and chemokines, etc. Okay, so these are very important. Okay, so these are very important. Humoral immunity is uh, based on the B lymphocyte because it releases antibody. So that's why we can say that that is the humoral and not the cell mediated, because B cell itself not fight to the pathogen. It releases some chemicals and that will go to the pathogen and fight with those pathogens. Okay, much more antigen specific immunity is the adaptive immunity because it is very specific. Okay, specificity means specificity. It means they are not general. Okay, they are not generalized. Means suppose a bacteria which is inserted into our body. Okay, and our body is thinking that which type of bacteria is, and we have to make the different type of chemical which is very specific to only this type of bacteria and not affecting the another type of bacteria, and then we have to kill them. So it is the specific. Okay, but whenever they are releasing some another type of chemicals which are not specifically for only one type of molecules, and for these are also using for the another type of bacteria also, and they are not discriminating between the or among the bacteria or another pathogens. Okay, means they following the same common pathway and the same common type of uh, activity uh, against the all the type of pathogens. So it means these are generalized. Okay, or we can say that these are not specific type of immunity, and we already know that non-specific type of immunity is involved under the innate immunity, which we already discussed in the immunity innate immunity video. Okay, so you can go and check there. Now coming to back that adaptive immunity, because as we discussed that it is very antigen specific. It uh, uh, it uh, it is it involved lymphocytes, and lymphocyte always wait for the signal that where the antigen is present in the cell and which type of antigen is there, or which type of pathogen is there. So they always wait for those all signals. So therefore it takes a lot much time, and that's why we can say that it takes uh, some days, about 5 to 6 days after the barrier breach. Whenever barrier is breaching, and barrier is considered under the innate immunity, which we discussed, that anatomical barrier, chemical barrier, right? So whenever they breaches, then it reaches to the adaptive immunity, because second option we have, that is the adaptive immunity. Whenever in our innate immunity is completely not working against all of them, and still we require some more type of immune booster, then adaptive immunity comes there and work against them. Okay, so that's why adaptive immunity comes there and work against them. Called as acquired, which I discussed to you. Okay, so it gives some booster effect to us. Okay, so this booster effect uh, give, given by the this type of adaptive immunity is released whenever pathogen is exposed towards our lymphocytes, and these uh, pathogens uh, are recognizing by the our APC cells, and whenever they or our phagocytes, and whenever the phagocytes and APC cell present their parts of antigenic part of those pathogens, their antigenic parts are presenting to the lymphocyte, then lymphocyte start working against them. Okay, so this is the exposure activity, and whenever first time this pathogen comes into our body, then it is called as first exposure. Means first time whenever you get diseased by that type of pathogen. Okay, so that is first exposure. Okay. So in the first time it takes a lot much time, because it uh, take time to get the signal, and then it rearrange their genomic structure, that they may be able to make that type of specific type of antibodies against them, or otherwise specific type of cells should be released and chemicals should be released, so that they can fight with those pathogens. Okay, so it takes some time, and that is 5 to 6 days almost nearby, and whenever it exposes into our body second time, that time our immune system already be ready, because in the first time whenever the exposes uh, whenever pathogen exposes to our cells, then they start proliferating and dividing. Then in that time they also make some memory cells, and these memory cells will help in the second time exposure, or we can say that in the second time response. Okay, so every cell, suppose this is the lymphocyte. Okay, whether it is B lymphocyte or whether it is T lymphocyte, whenever it start responding against the pathogen, then it always releases two type of cells. One which will fight to pathogen. Okay, and second it will make memory cells. Okay, so it will make memory cells, so it will in help in the second time response or second exposure. So it will help in second exposure, but that was the first exposure, right? So in this second exposure, our body will be ready that time. Okay, so that in the second time, uh, this adaptive immunity will take very few or very less time, and it will start fighting to the pathogen very quickly. Okay, so that is the benefit. Whenever the second time pathogen comes into our body, then we are already ready that time. Okay, so the slower, because wait for the antigen specific and presenting cell, as I told you that it is based on the antigen presentation. Whenever the cell comes, any cell comes to it, and they give some signal about the antigen, then it start working. Otherwise it will wait for the antigen, and that whenever the antigen will come, and they start working against them. Okay, so that's why it takes some time, and that's why it is very slower. And after antigen encounter, T and B lymphocyte undergo the selection and proliferation process. What is the selection and what is the proliferation? So always these all cells, like suppose that is the B cell, or suppose that is the E cell, all the lymphocytes are producing in the bone marrow, and after the bone marrow whenever they producing, that time they are not able to recognize any person. Okay, so they have to gain the activity, they have to gain the this type of training, and that's why that is called as selection process. So in the selection process they are, are trying to identify that is this cell which is created now a time, so is this able to recognize the person or not. If they are recognizing and if they are able to recognize, it means that time they will be selected for the next process and sent to them in the affected area and start proliferating. Otherwise, suppose that is not able to identify the person accurately, it means they are not useful for us, and these this start apoptosis and will be degraded. Otherwise, they will be discarded from our body by the some destruction processes of this of those cells. Okay, so that's why it is called as selection. And after the selection, whenever they are sending to the uh, uh, pathogen infected site, then they go there and start proliferating and uh, makes many more cells. Okay, so that is the process, and we will discuss about the selection in very brief in this topic in this video, and in detail if you want, then please wait for some time whenever we will discuss about all these separately, then I will make another video in which complete B cell and T cell will be uh, discussed, that how they start working and how they which type of activity they have and what are the process by this by which type of process they start uh, killing or start blocking the pathogens. Okay, so that time for that time please wait, and now a time we are discussing in brief. Okay, so adaptive immunity, and I am giving the example of the adaptive immunity like vaccine against the infectious disease. So suppose when vaccine is provided to a host, and that is the uh, working against the pathogen, so that is the okay, so suppose it works against the pathogen, so it means that is very important, and this is creating some specific type of activity again in our body against the pathogens. Okay, so because activity is created or activity is generated by the motivation from the outside, so that's why it is called as adaptive, because we adapt, we are not having, we are just adapting from the outside environment. So that's why it is called as adaptive immunity, or we can say that acquired immunity. Now coming to the its main important parts, and these are the lymphocytes, as I told you in the cellular part, B lymphocyte, T lymphocyte. Okay, so these are some cells. So I'm talking about the lymphocytes. So there are two types of lymphocytes: B lymphocyte and T lymphocyte. B lymphocyte which mature in bone marrow, and in case of birds, there are another organ that is bursa fabricus, and in that they goes after the making on the bone marrow, they goes to there and get some training about this type of specificity. Okay, and in T cells which mature in the thymus, but already they uh, uh, originated in the bone marrow. Okay, so origin for both these cells are bone marrow, originate in bone marrow always. Okay, but then they go to the either bone marrow or to the in case of birds to the bursa fabricus in case of B lymphocytes, but in case of T lymphocyte, the T lymphocyte goes to the thymus. Okay. So both of these B and T lymphocyte are distinguishes based on the cluster of differentiation, that is called as CD for cluster of differentiation, and there are lots of CD, almost about 300 or more than 300 CD has been discovered, and still uh, they are assuming that they are in higher number. So there are lots of CDs available on the surface of these cells, and by the help of these CD, they work or they give us immunity. How they give us immunity, we will discuss about all these things later on. Okay, there are some specific type of proteins which which are also using for the identification of the T cells. Now coming to the one by one these cells. Okay, so first of all, the B cell. B cell are the or B cell or B lymphocyte are those cells which are using in the humoral immunity. Okay, so these are involved in the humoral immunity, which I already told you. Okay. These B lymphocytes are 5 to 15% of the lymphocytes in the blood, and rest all the lymphocytes are T lymphocytes. So we can say that T lymphocytes are higher, present in the bone marrow, spleen, lymph nodes, and mucosa associated lymphoid tissues. Okay. So because as we discussed in the previous video that lymphocytes are all mostly present in the lymph node, so that's why these are also present in the lymph node, and whereas they are also present in the bone marrow and spleen too. Functions of the B lymphocytes are B lymphocytes are the simple type of lymphocyte, and they start working whenever pathogen or antigen encounters by these cells. Okay, so whenever they identify that there is pathogen, and after the identifying, they start processing. Okay, they start processing and they divide into two type of cells. As I told you in the above, that every cell divided into two type. One is memory cell and another is in the fighting cells. So it's a memory cell. Okay, so these are B memory cells, and these are the plasma cells. Okay, plasma cell are the larger cells, and they start releasing chemicals, and these chemicals are antibodies. Okay, so they start releasing these type of chemicals, and, and these all chemicals are the antibodies, and these antibodies go to the pathogen and kill them or precipitate them. Okay, so these all activity are done by these type of antibodies, or antibodies are simply called as immunoglobulins or IgG. Sorry, IG. Okay, there are lots of type of IG which will be discussed in this video later on. So functions and functions of the this B cell are the plasma cell, which produces or manufacture the antibody and then secrete them. And in case of memory cell stores some memory, and these B cell also able to present antigens to the T cell and release or start the releasing cytokines by the help of or through the T cells, and they signal for the releasing of cytokines to the T cells are given by the B cells. Okay, so these type of antibodies are having two type of feature. Okay. In this one case, it will be rely on the surface of its B cell. Okay, otherwise they will be released to the blood. So it might be in the open form or free form. Otherwise, it might be present on the surface of the B lymphocyte. Okay, so there are two type of its fate: formation and maturation of the B lymphocyte. B lymphocyte always originate in the bone marrow, and these are called as pro B cell. It converted into pro B cell, and then pro B cell converted into the immature B cell. Immature B cell have some immune tolerance. At checking the immune tolerance, that it means are they able to recognize the pathogen and recognize the antigen or not. If they are not recognizing the antigen, then apoptosis will be occur in those cells and kill those non-identifying cells. Otherwise, those type of immune cells which are uh, increasing the autoimmunity or autoimmune type of diseases, then these cells also will be killed or also will be discarded from our immune system. And after this complete selection, this is called as immune tolerance, and after this selection it will convert it into the immature B cell. Okay, so that is the selection. That was the selection, and after the selection it converted into the immature B cell, and immature B cell again converted into the mature naive B cell. So now these mature naive B cell goes after maturation, it goes to the another type of tissue which are present in the peripheral lymphoid organ, or otherwise maybe in the another or tissue. Okay, so they leave the bone marrow and go to the peripheral lymphoid organ, and they start encounter antigens. They are ready to encounter the antigens, and this is called as immune response. So there are two type of immunological terminology. One was the immune tolerance, and second is the immune response. So you can understand both of these concepts. Immune tolerance is the process in which the B cell or T cells are recognizing and identifying that are they able to recognize the pathogen or antibody or not, sorry, antigen, or if they are not identifying the antigens and they start working against our body's antigen itself, so it means they are harmful to us, and this type of cell will be discarded, and rest all the cells will be keep safe and will send to the further process. Okay, so that was the immune tolerance, and the immune response, whenever they encounter the antigen, then start working to stimulating its immune system, and this is called as immune response. So B cell uh, types and steps of the immune response. Okay. B cell, as I told you, that they start the immune response, and there are two types of immune response: primary immune response and secondary immune response, which I already told you that first response, which is called as primary immune response, and second time whenever we are attacked by the pathogen, that is called as second immune response. So second immune response is the anamnestic or booster immune response. Sorry, I want to uh, pardon me. Actually, I did a mistake. Actually, I told you that anamnestic or booster type of immunity. So I am erasing from here, and that is the actually second immune response. Okay, not the adaptive immunity. I'm correcting myself. Pardon me. Okay, so that is the anamnestic type of or booster type of immunity or immune response, which is comes in the second time immune response. Okay, T cells. T cells are those another type of lymphocytes which also work for the our immune system or acquired our adaptive type of immune system, and this gives us the cell mediated immunity. Okay, so T cell gives us the cell mediated immunity, and T cells originate and transfer. Where originate and transfer? As I told you, that they develop in the bone marrow stem cells, develop from the bone marrow stem cells, and they develop into bone marrow. And then after the bone marrow, they travel to the thymus, and thymus giving them training and on the basis of the selection. So this is the again selection process as was happen in case of the B cells. Okay, so that is this selection process, and in this selection process they are checked for the immune tolerance and also for the autoimmune diseases. Okay, or either they are identify MHC and antigen or not. Okay, suppose they are identifying their itself MHC and itself and antigen recognizing receptors, then it means they are not useful for us, and these type of cells will be discarded or degraded, and rest cells will be uh, go for the next process, and that process is called as the selection process. In this selection process, the lower grade cells or the cells which are not viable or which are not useful for us will be discarded, and rest all cells will be selected for the further process and giving training to them, like how to fight against the pathogen, and they working on this process and they fight to the pathogen. Okay, so selection is then occur in the thymus, and after the selection, the T cell can recognize the non-self antigen complex to self MHC molecule or not, as I told you. Okay, so after this complete process, they converted in, they are living for the living, and that is the survival. And after the survival, they leave the thymus because now a time training has been completed, and then they leave the thymus and go to the peripheral blood and lymphoid tissues. Okay, so in these tissues they live there and wait for the signals. Whenever they come, they receive the signal which is coming from the NA cell, and then start giving signal to the another type of cells, or either or otherwise, and they will start differentiating into the another different cells, and these different cells will work against those pathogens, and either they kill or either they uh, work against those, like in another way, like in complement system, or there are lots of pathways, and we will discuss about all these things later on. Okay, so these T cells have some receptors also, and on the basis of these receptors we start identifying these cells, and these receptors are CD4 and CD8. These receptors are very important. Actually, there are TH cells which have CD4 type of cluster of frequencies on those cells. Okay, and another cells, these are the C cells or cytotoxic C cells. They have CD8 type of cell surface receptors are there. Okay, so there are two type of cell surface proteins are present, and on the basis of these specific protein we can identify that okay, these are the TH cell and these are the TC cells. Actually, TH cells commonly or usually have CD4, but sometimes may also have CD8 on the same cell. but mostly see, they have CD4 and TC have CD8. Okay, so this is the clear-cut differentiation. T cell receptors are also present there. These are called as TCR, and these TCR have two type of chain. One is alpha beta chain, and one is alpha beta type of protein or TCR. Okay, and second is the gamma beta, gamma delta type of TCR. And these names are given on the basis of its chain. If they have alpha chain and beta chain, then these type of TCR are called as alpha beta TCR, whereas another which contain the gamma and delta type of chains, these are called as gamma delta TCR. Okay. Now coming to the functions of the T cells. Functions of the T cells are two signal model for this T cell activation, and which will be discussed in the T cells later on, that which is the first signal and which is the second signal. Okay, so we will discuss about this process in very detailed manner later on. Now I am giving only brief about the adaptive immunity, so you can understand the flow chart of that what is the adaptive immunity. Okay, uh, T cell again have some different types, and these are called as T helper cell, T regulatory or suppressor cell, T cytotoxic cell. Okay, so these are different type of T cells, and these T cells have their own specific types also. Like T helper cell, they have CD4 and they differentiate into the Th naught cell, which again convert into any one of them. It might be into Th1 cell, it might be into Th2 cell, or it might be into Th17 cell. Okay, so there are lots of type of Th cells, and these are the specificity. This is the specificity of the T cells and T lymphocytes. That's why we are saying that there is another type of immunity, that is the cell-mediated immunity. Okay, because it differentiated into the many type of cells, and we will discuss all these cells, that what is the main function of Th1 cell, Th2 cell. Okay, so all these will be discussed later on. So this was the cellular part of the adaptive immunity. Now coming to the molecular part. Okay, so there were two type of cells important cells, T lymphocyte and B lymphocyte. Okay, now coming to the its molecular part of the adaptive immunity. Molecular part contain the cytokines and antibodies. As I mentioned here, cytokines are the polypeptide and they themselves are not antigen specific. Okay, so that is the very important. And cytokine connect an innate and acquired immunity, which is also very important, because whenever we are talking about the innate immunity and then again talking about the adaptive immunity, so do you know that are all these systems are connected? Okay, because they need each and every cell. As I told you that adaptive immune cells like B lymphocyte and T lymphocyte wait for the signal that any cell come towards them and give the antigenic signal to them, then they will start working. So which cells give them signal? Actually these cells are the innate cells. Okay, so innate immunity cells of the innate immunity give some signals to the adaptive immunity, and then adaptive immune cells start working. Okay, so every part of the immunity is connected with each other. So that's why in this case we can say that cytokines connect the innate and acquired immunity. How they connect, we will discuss it later on, because we have complete chapter on this cytokine. So I will make some videos on these also. So please wait while they will be released. Play role in the inflammatory responses. It is also important part, because as I told you that there is suppose redness, there is pain, there is uh, sometimes heat heated part of our body. So that is some signs of the uh, inflammatory response. Okay, so inflammatory response is also reached by the cytokines. So that is very important. Cytokines are secreted by the immune and other cells. So these are secreted, and when the cell interact with the specific antigen. Okay. So whenever pathogen or pathogen associated molecules comes toward them, and then they suppose that is endotoxin or other type of any molecule, then they come towards them there, and then cell releases some chemicals, and these are called as cytokines. Actually, cytokines have lots of type of cytokines, and they contain it might be TNF. Okay, might be IL type, it might be chemokines. Okay, so there are lots of types of cytokines which we will discuss, that what are the types of all these chemicals and how they start function. Okay, so these are another types of chemicals. Okay, lymphocyte triggers cytokine secretion. So lymphocyte can also trigger, as I told you, that immune cell, whenever they interact with the specific antigen, then they secrete these are chemical, these cytokines. So in the same case, lymphocyte, which are very important in the case of cytokines, because whenever we will discuss about all the cytokines which are released by the specific cells, then we will discuss that how it started working and how they encounter the pathogen and how they cover up those all the antigens surround them. Okay. Now coming to the antibodies. Antibodies are specific glycoproteins. Glycoproteins means they are containing sugar plus proteins, or we can say that sugar plus peptide. Okay. So that's why these are called as glycoproteins, glyco, sugar, and proteins. Okay, so they contain these type of molecules, and they are also called as immunoglobulins or Ig. Okay, so these are called as immunoglobulins, or we can say them Ig in short form. Okay, so Ig are antibodies actually. So what are the function of the antibody? Antibody always receive the antigen which are moving in the uh, our body whenever they comes in our body. Okay, so antibody which are releasing in our body, and they are coming there, and they are trying to find out that where antigen has come. Okay, so antigen receptors are present on these antibodies. Okay, so antibodies and antigen fit tightly together because their shape and surface proteins like charge and are complementary. Okay, so they have a specific type of uh, its structure, and based on these structure we can say that antibody are very specific, and they contain some parts here. Okay, and this is, suppose, this part is its variable region. We will discuss in later on matter that in later on videos, that what are the variable region and what are the constant region. Okay, so these sorry. Okay, so this is the part called as variable region. Okay, this also, and rest complete part is called as constant. This is constant region, and they also contain two type of chains, important chains. First one was like this, this is the second. So these are the heavy chain. Okay. These are the heavy chains, and they also contain some light chain, and these are called as light chain. So these are light chain. This is also again light chain. So they contain two heavy chains and two light chains, and these all are connected with each other. Okay. With these all are connected with the disulfide bond. Okay. So these are connected with the disulfide bonds, and that's why they are very specific and unique. And these antigen have their their they have specific sites on these, and these sites are site to bind with an antigen. Okay, so they always bind antigen here and here. Okay, so variable region can convert and can change itself into any type of sequence, and so that they can uh, join with the antigen. Okay, and if there is any type of antigen, so we can fight against them, and we can uh, acquire some immunity against them. Okay, so that's why the variable region are very important, and this is the common structure of a typical antibody. And there are lots of another type of antibodies also there. Okay, like these antibodies are divided into many important 5 classes, and these are IgM, IgG, IgA, IgD, IgE, M for mu, okay, G for gamma, A for alpha, D for delta, uh, okay, D for delta, and E for epsilon. Okay, so all these are very important, and these all antibodies are secreted by the plasma cells. As I told you that B cells convert into two cells, one divided into two cells. One is the memory cell and one is the plasma cell, and plasma cells start secreting the antibodies. Okay, so that's why the plasma cell are very important for the secretion of the antibody, and they are present in blood flow or on the surface of the B cells, as I told you in previous in the above point also, that they might be present on the B cells, and they can be released from the B cell from the plasma cell uh, into the blood flow. Okay, so uh, they might be present in the blood flow, and they might be present upon the surface of the B cells. Okay, and there are 5 major classes, as I told you, and they also contain lots of different types under these classes also. Okay, so we will discuss all about all these things later on. Okay, that how the antibody works, and which type of antibody are very important. Okay, and whenever in primary response which type of antibody releases, and in secondary response which type of antibody releases, and why. Okay, so this is the our complete topic. This was the about adaptive immunity, and hope you have gained all these things very clearly. If you have any doubt, then please let me know. Okay, thank you so much.